purification. DNA can be prepared from a whole range of materials including blood, exfoliated bladder cells, frozen tissue samples, and paraffin tissue blocks. Since DNA is generally stable under suitable storage conditions, it can be prepared and stored in batches. DNA extraction and purification involves three basic steps: breaking the cells open to expose the DNA, removing the membrane lipid through the use of an appropriate detergent, and precipitating the DNA with alcohol. There are also two optional steps that are almost always done prior to the precipitation step: the removal of protease by adding an appropriate protease, and the removal of RNA with the use of an RNase. Through DNA purification, our samples, you can minimize the disturbance from non-target DNA and other contaminants and increase the stability of your sample in long-term storage. RNA extraction and purification, on the other hand, involves four basic steps. First, you need to distribute the cells by adding vanadium thiosinate and the reducing agent to the sample and the subject into vigorous shaking or vortexing. This step will also break the distal head bonds and inactivate the contaminated proteins present in the sample. After cell lysis, you need to add phenol and chloroform isoamyl alcohol to separate your RNA sample from the solution. The echo phase contains your precious RNA, so you need to put it in a separate tube. Add isopropanol and centrifuge the solution to precipitate your RNA. Washing the precipitate with 75% ethanol will remove any impurities from your sample. However, it is important to note that RNA is less stable as compared to DNA, so you should only isolate it prior to use. Special care should also be taken before, during, and after the process to ensure the purity of your sample. Remember, even the smallest amount of RNase can compromise its quality. DNA and RNA can also be isolated from the same biological sample by extracting a total nucleic acid fraction and dividing into two parts, one of which will be treated with DNase, while the other portion will be treated with RNase, to recover RNA and DNA respectively.
Protocol for DNA extraction. 700 microliters of human blood were added in a microtube and also 800 microliters of sterile water in order to reach 1.5 milliliters. It was then mixed by inversion and centrifugated at 13,500 rpm for 1 minute. Then 1 milliliter of the supernatant was discarded. And then we added 1 milliliter of sterile water and then it was mixed by inversion. Next, we centrifugated at 13,500 rpm for 1 minute and then we discarded the supernatant. We added 375 microliters of sodium acetate, 0.2 molar, 25 microliters of SDS-10X, and 15 microliters of proteinase K to the sample. Then the sample was resuspended briefly with the vortex. Then the sample was incubated at 37 Celsius for 1 hour and 8 minutes. The sample was briefly centrifugated in order to make a deposition of all the condensates generated. 600 microliters of phenylchloroform isoamylic alcohol were added, and then it was mixed by inversion for about 20 seconds. Then the sample was centrifugated for 2 minutes at 13,500 rpm. The aqueous phase was recollected in another microtube. As we obtained 440 microliters of the sample, 2 volumes of absolute ethanol were added, which means that 880 microliters of it were added. Then we centrifugated for 5 minutes at 1500 rpm, and the supernatant was eliminated. Next, we added 48.88 microliter of sodium acetate, 3 molar, in order to resuspend the pellet. Next, the sample was incubated in water bath at 15.5 Celsius for 10 minutes. 5,000 microliters of ethanol were added. It was mixed by inversion and centrifugated for 10 minutes at 13
Quantification by spectrophotometry. A 1 to 100 dilution was made with the DNA and the RNA resuspended pellets in order to obtain 1 milliliter of each sample. That means add 10 microliters of the DNA or RNA sample and then add 990 microliters of distillated water. The spectrophotometer was turned on and set. Then 1 milliliter of water was put into a spectrophotometer cell in order to set the target. Then the 1 milliliter dilutions of DNA and RNA were put into spectrophotometer cells. Both were read at 260 and 280.
Here we have the results shown in the spectrophotometer. We can see the absorbance at 260 nanometers of the human blood that was 0.088 nanometers, and the absorbance at 280 nanometers was of 0.050 nanometers. The concentration of the sample was 454 micrograms per milliliter. The ratio between the absorbances of 260 nanometers and 280 nanometers for the human blood sample was 4.4054 micrograms per milliliter. As we resuspended 40 microliters of water in the sample, we multiplied those values and then we make the relationship between the ratio that was 1 to 100%.